The overall goal of this procedure is to demonstrate techniques for encapsulating cells in thiol-ene photopolymerized hydrogels, as well as subsequent recovery of naturally formed beta cell spheroids from the enzyme-sensitive hydrogels for biological characterization. This is accomplished by first mixing the Min6 beta cells with a 4-arm PEG-norbornene macromer and an enzyme-sensitive peptide crosslinker. The cells are then encapsulated using free radical-mediated thiol-ene photopolymerizations to form a hydrogel. The second step is to culture encapsulated cells in thiol-ene hydrogels in a carbon dioxide incubator at 37 degrees Celsius. After the cells have proliferated to form aggregates, the cell-laden hydrogels are rinsed with buffer solution and then incubated in chymotrypsin solution under gentle shaking for gel erosion. Then, the recovered aggregates are carefully washed to remove enzyme solution and eroded polymer. Ultimately, these cell aggregates can be used for structural and functional analysis.
So the main advantage of step growth thiol-ene photochemistry is that its reaction conditions are milder than the traditional chain growth PEG diacrylate. Therefore, the thiol-ene hydrogels provide an excellent microenvironment for many cell types, including pancreatic beta cells.
The implication of this technique extends towards various cell-based therapies because this thiol-ene system provides an excellent microenvironment for in vitro experiments and a great platform for cell delivery. While we synthesize the peptides used in these experiments in our lab, there are many companies from which the peptide crosslinkers we described can be obtained.